Protect your hearing. Recommended safe noise levels to prevent hearing loss. Noise doesn't rest, the quiet menace threatening our hearing. In a world that seemingly never stops, a silent epidemic is quietly plaguing our auditory health. The culprit? Noise. The unwanted and harmful sound that permeates our daily lives. As a leading science journalist, I've delved into the latest research, and the findings are both alarming and enlightening. For decades, experts have known that exposure to excessive noise leads to noise-induced hearing loss, NIHL, a debilitating condition that can severely impact communication, social interactions, and overall quality of life. But the true safe level to prevent this damage has remained elusive, until now. Recent studies suggest that the safe noise exposure level may be far lower than previously thought. Groundbreaking research by pioneers like Dr. Carl Kreiter has uncovered that the threshold for reversible auditory damage, the effective quiet level, is around 55 to 60 decibels, dB. This is a radical departure from the current occupational limit of 85 decibels and the EPA's public guideline of 70 decibels. The implications are staggering. Everyday sounds we take for granted, from city traffic to power tools, may be slowly chipping away at our hearing. And the problem starts early, with children and teenagers exposed to dangerously high noise levels from personal devices and recreational activities. But this isn't just about occupational hazards or noisy weekends. The true culprit is our modern lifestyle, where silence has become a rarity. The relentless cacophony of industrialized societies, from construction to air travel, has become the backdrop to our daily existence. And the damage is accumulating, with hearing loss no longer just an age-related condition, but a direct result of a lifetime of noise abuse. The good news is that NIHL is entirely preventable. By understanding the actual safe noise levels and redefining how we think about noise, we can empower individuals and communities to protect their auditory health. This means advocating for stricter noise regulations, promoting noise-reducing technologies, and fostering a cultural shift that values the restorative power of quiet. It's time to reclaim our acoustic environment and safeguard our most precious sense. As we navigate this noisy world, let's remember that the true mark of a civilized society may be its commitment to preserving the delicate symphony of silence. Source: www.nature.com. Unlocking the power of membrane-embedded machinery. Diving into the intricate world of membrane proteins, unlocking the secrets of cellular communication. In the ever-evolving landscape of scientific discovery, the study of membrane proteins has emerged as a captivating frontier, shedding light on the intricate mechanisms that govern cellular communication and function. As a science journalist deeply immersed in this field, I am thrilled to share with you the remarkable advancements that have unfolded in this realm. Nearly 4 decades ago, the first membrane protein structure was unveiled, marking a pivotal moment in our understanding of these molecular marvels. Since then, the pace of progress has been nothing short of astonishing, as researchers have delved deeper into the complexities of these integral players within the cellular landscape. Membrane proteins are not mere passive bystanders, they are the unsung heroes orchestrating the intricate dance of compartmentalization, transport, and signaling within the cellular realm. From the exchange of water, ions, and larger molecules to the propagation of neural impulses, these molecular machines are the linchpins that enable seamless communication across the boundaries that define our cellular universe. The breadth of their functions is truly astounding, as evidenced by the sheer diversity of roles they undertake, from serving as receptors to facilitating active or passive transport, from acting as cargo receptors in vesicular trafficking to playing pivotal parts in membrane protein biogenesis. The versatility of these membrane-embedded wonders is a testament to the ingenious design of nature's engineering. Yet, despite their ubiquity, the progress in unraveling the mechanisms underlying their action has not always kept pace with the advancements in the study of soluble proteins. Until recently, the structural determination of these elusive entities has been a considerable challenge, owing to their preference for a hydrophobic environment within the lipid bilayer. However, the tide has turned, and the current issue of Nature Structural and Molecular Biology showcases a remarkable collection of papers that delve into the intricate workings of membrane proteins. From the revelations about the activation mechanisms of ion channels and transporters to the insights into the conformational changes that drive signaling cascades, these studies have pushed the boundaries of our understanding. The work of Kaliankova et al., for instance, has shed light on the basis of peptide activation in excitatory DEG-ENAC channels, while the research by Fortier and colleagues has unraveled the activation mechanism of the prototypic Escherichia coli CLC type Cl-H+ exchanger. These findings not only expand our fundamental knowledge but also hold the potential to inform the development of targeted therapies for a range of ailments. Moreover, the field has witnessed a shift in focus, with researchers diving deeper into the mysteries of orphan receptors, those with unknown ligands. The insights provided by the studies on GPR 161 and GPR 156 have not only advanced our conceptual understanding but have also opened new avenues for therapeutic exploration. Beyond the realm of receptors, membrane proteins play pivotal roles in energy production, as showcased by the work of Sharma et al., which has shed light on the elastic coupling mechanism of ATP synthase. This knowledge is crucial in our quest to harness the power of cellular machinery for sustainable energy solutions. As I delve into the pages of this remarkable issue, I am struck by the sheer breadth and depth of the discoveries unfolding in the study of membrane proteins. From the elucidation of extracellular receptor assemblies to the insights into chemokine receptor and serotonin 3 receptor agonism, the scientific community is leaving no stone unturned in its pursuit of understanding these cellular powerhouses. The future holds immense promise, as the next 30 years of progress in this field are poised to unlock even more profound secrets. With each new structure, each novel mechanism, and each innovative application, the world of membrane proteins continues to captivate and inspire, pushing the boundaries of our scientific understanding and paving the way for transformative breakthroughs. Source: www.nature.com. Exploring key factors influencing worldwide tree variety, unraveling the mysteries of global tree diversity. In the ever-evolving landscape of our planet, one of the most captivating biodiversity patterns is the decline in plant species diversity from the equatorial regions to the poles. A recent study published in Nature Plants sheds new light on the intricate mechanisms driving this remarkable latitudinal gradient. The research, led by a team of ecologists, delves into the role of local neighborhood interactions among tree species, a concept known as conspecific density dependence, CDD. The Jansen-Connell hypothesis posits that host-specific natural enemies, such as pathogens, parasites, and herbivores, are more prevalent in tropical forests, favoring the growth and survival of heterospecific, different species, trees over conspecific, same species, trees. This, in turn, is thought to maintain the high levels of plant diversity observed in the tropics. However, the findings of this latest study suggest a more nuanced understanding of this long-standing hypothesis. The researchers analyzed data from 23 stem-mapped forest plots across the globe, spanning a wide range of latitudes. Their analysis revealed that, on average, the phenomenon of stabilizing CDD, where increases in conspecific density reduce survival more than increases in heterospecific density, is indeed present across these forests. Interestingly, while the researchers did find a slightly greater average stabilizing CDD in tropical compared to temperate forests, this relationship with latitude was not statistically significant. This unexpected finding challenges the traditional view of the Jansen-Connell hypothesis as the primary driver of the latitudinal diversity gradient. But the true gem of this study lies in its examination of stabilizing CDD patterns for tree species at different abundance levels within their communities. The researchers discovered that stabilizing CDD is significantly stronger in tropical than temperate forests for species at low to moderate abundances. This finding supports a more nuanced interpretation of the Jansen-Connell hypothesis, suggesting that differences in stabilizing CDD for less common species may play a crucial role in shaping global diversity patterns. The concept of natural enemies as stabilizing forces may seem counterintuitive, but the researchers propose an intriguing mechanism. As host populations decline, their host-specific enemies also decrease, allowing the host species to rebound and stabilize at relatively low to moderate abundances. This process, if more pronounced in tropical forests, could enable the tropics to serve as a museum of plant diversity, preventing the extinction of species at low to moderate abundances. The implications of this study extend beyond the Jansen-Connell hypothesis, as it highlights the need for a deeper understanding of the multifaceted drivers of global biodiversity patterns. The researchers suggest that examining stabilizing CDD across different life stages, from seedlings to adults, may provide further insights into the complex interplay between local interactions and global diversity gradients. As we continue to grapple with the alarming rates of biodiversity loss worldwide, studies like this one offer valuable clues to unraveling the intricate mechanisms that underpin the remarkable richness of life on our planet. By uncovering the nuances of local interactions and the global scale implications, we may unlock new pathways to safeguarding the precious diversity that makes our world so vibrant and awe-inspiring. Source: www.nature.com. Unlocking macrophages, releasing inhibitory signals to clear cell debris, unraveling the secrets of macrophage efficiency, releasing the proximal brake for efferocytosis. In the ever-evolving world of cellular biology, macrophages have long been recognized as the unsung heroes, tasked with the crucial role of clearing out cellular corpses and debris. These versatile immune cells have demonstrated remarkable efficiency and responsiveness, yet the intricate mechanisms underlying their rapid response to environmental stimuli have remained largely elusive. That is, until now. A groundbreaking study published in Nature Structural and Molecular Biology has shed new light on the inner workings of macrophages, unveiling a novel regulatory mechanism that allows these cells to execute efferocytosis, the process of engulfing and degrading dead cells, with remarkable precision and speed. The study, led by T2 Van and colleagues, delves deep into the transcriptional landscape of macrophages, revealing a remarkable shift in gene expression patterns upon encountering apoptotic cells. The researchers discovered that several genes, which were previously held in a paused state by the enzyme RNA polymerase II, POL2, suddenly overcome this roadblock and become rapidly upregulated. The key to this transformation lies in the activity of the kinase CDK9, which is responsible for the release of the paused POL2. By inhibiting CDK9, either through generic or specific inhibitors, or by degrading the enzyme using specialized proteases, the researchers were able to disrupt the efficient efferocytosis performed by both mouse and human macrophages. Interestingly, this regulatory mechanism appears to be specific to efferocytosis, as other types of phagocytosis, such as Fc receptor-mediated phagocytosis, remained unaffected by the POL2 pause release inhibitors. The researchers also uncovered a pivotal role for the transcription factors EGR3 and EGR1 in this process. Upon encountering apoptotic cells, macrophages rapidly upregulate the expression of these genes, both at the mRNA and protein level. Depletion of EGR3 attenuated efferocytosis, while overexpression of the same factor conferred partial resistance to the inhibition of POL2 pause release. Further investigations in zebrafish embryos revealed that EGR3-deficient microglia, the macrophage-like cells in the brain, were significantly less proficient in engulfing apoptotic neurons, both during the initial encounter and in continuous efferocytosis. The study also uncovered a network of EGR3-regulated genes that are closely linked to the endosomal and lysosomal pathways, as well as the cytoskeleton, suggesting that this transcriptional reprogramming plays a crucial role in the functional aspects of efferocytosis, such as phagosome acidification and cytoskeletal remodeling. This groundbreaking research not only advances our understanding of how macrophages clear apoptotic cells but also highlights the unexpected regulatory role of POL2 pause release in this process. The identification of EGR3 and EGR1 as key effectors of the transcriptional response opens up new avenues for investigating the potential involvement of these transcription factors in other immune cell functions and the potential implications in neuropsychiatric disorders. As we continue to delve deeper into the intricacies of cellular biology, studies like this one serve as a testament to the power of scientific inquiry, reminding us that even the most well-studied systems still hold untapped secrets waiting to be uncovered. Source: www.nature.com. Exploring the faces of science, 50 captivating photographs for Londoners, unraveling the kaleidoscope of science, a photographic journey through the lives of exceptional researchers. In a captivating display that shatters preconceptions, Londoners are now granted a rare glimpse into the diverse and extraordinary world of scientific research. The Where I Work photo exhibition, a collaboration between Nature and Argent, brings to life the stories of over 50 pioneering scientists, showcasing the remarkable individuals behind the groundbreaking discoveries that shape our understanding of the world. Adorning the bustling King's Cross area, these stunning portraits challenge the traditional notions of what a scientist looks like and what they do. From a medicinal plant researcher in Jamaica to a biotechnologist exploring the genetics of ornamental flowers, the exhibition celebrates the boundless creativity and intellectual curiosity that drives these remarkable individuals. The images are a refreshing departure from the stereotypical depictions of scientists, remarks Sarah Naylor, a local resident. Seeing these diverse and inspiring individuals at work is a powerful reminder that science is not just a pursuit for a select few, but a dynamic field open to all. The exhibition, dedicated to the memory of Karen Kaplan, the visionary behind the Where I Work initiative, serves as a testament to the tireless efforts of those who have dedicated their lives to advancing our understanding of the natural world. Kaplan's passion for showcasing the human face of science has now found its tangible expression, inspiring a new generation to consider careers in the STEM fields. As I walk through the exhibition, I'm struck by the sheer breadth of expertise and the human stories behind these images, says Tom Wilkins, a university student. It's a powerful reminder that science is not just a collection of facts, but a living, breathing pursuit driven by the curiosity and determination of remarkable individuals. The Where I Work exhibition will remain on display until June 2024, offering Londoners a unique opportunity to connect with the scientific community and challenge their own preconceptions. Through these captivating portraits, the exhibition aims to inspire the next generation of researchers, innovators, and problem solvers, who will undoubtedly shape the future of our world. Source: www.nature.com. Eternal Chaos: Io's History of Violent Volcanic Eruptions. Unleashing the Secrets of Io, a Volcanic Moon's Billion-Year History. Nestled within the vast expanse of the Jovian system, Jupiter's moon Io has long captivated the scientific community with its relentless volcanic activity. Now, a groundbreaking study published in the prestigious journal Science has shed new light on the moon's tumultuous past, revealing that its volcanoes have been actively reshaping the surface for billions of years, potentially since the dawn of the solar system itself. The findings, meticulously pieced together by a team of planetary scientists, offer a unique window into the early conditions of our own planet, Earth, and the potential for life on other celestial bodies, such as the nearby moon Europa. Using the powerful Atacama Large Submillimeter Array, ALMA, in Chile, the researchers closely examined the sulfur-rich gases being ejected from Io's volcanoes. By analyzing the ratio of different sulfur isotopes in these emissions, they were able to uncover a remarkable story of the moon's long-term evolution. The data revealed that Io has lost a staggering 94 to 99% of its original sulfur over the course of billions of years. As the lighter sulfur-32 isotope is continuously stripped away from the top of Io's atmosphere at a rate of 1 ton per second, the overall composition of the moon's sulfur has become increasingly weighted towards the heavier sulfur-34 isotope. This discrepancy has accumulated over time, and by extrapolating the current rate of sulfur loss, we can conclude that Io's volcanoes have been actively erupting for the majority of the solar system's history, explains Catherine DeClear, a planetary scientist at the California Institute of Technology and a co-author of the study. The researchers' findings not only shed light on Io's turbulent past but also have far-reaching implications for our understanding of the formation and evolution of the Jovian system. The continuous volcanic activity on Io is driven by the complex gravitational interactions between the Moon, Europa, and Ganymede, as they orbit in a synchronized dance around the gas giant Jupiter. This orbital dance has likely been in place since the birth of the solar system, or shortly thereafter, DeClear notes. This raises the intriguing possibility that Europa, long considered a prime candidate for extraterrestrial life, has also been experiencing similar heating and geological activity for a similar duration, a factor that could greatly enhance the habitability of its subsurface ocean. Furthermore, the study provides a unique glimpse into the early stages of Earth's own volcanic history. The scorching, fluid lava flows observed on Io are akin to the magmatic processes that once dominated our planet's surface, laying the foundations for the continents and oceans we recognize today. Io's volcanism offers a window into the mechanisms of volcanism and Earth's early history, says DeClear. Understanding this extreme volcanic world can shed light on the formative years of our own planet, when massive lava flows and intense geological activity were the norm. As the scientific community continues to unravel the mysteries of Io, the implications for our understanding of the solar system and the potential for life on other worlds only grow more tantalizing. This latest study has not only revolutionized our perspective on the volcanic moon but has also opened up new avenues of exploration, promising to reveal even more about the dynamic and ever-changing universe we call home. Source: www.nature.com. Introducing Goldene, the gilded cousin of graphene. Behold the shimmering promise of Goldene, nature's latest marvel. In a remarkable scientific feat, researchers have unveiled the world's thinnest gold leaf, a mesmerizing material known as Goldene. This gilded cousin of the iconic graphene is a single atom thick, opening up tantalizing possibilities in fields ranging from catalysis to light sensing. The creation of Goldene marks a significant milestone in the quest to harness the unique properties of two-dimensional, 2D, materials. While scientists have long sought to synthesize atomically thin sheets of metals, the tendency of metal atoms to cluster together has posed a formidable challenge. But the ingenious approach developed by the Linköping University team has finally cracked the code, crafting a freestanding 2D layer of gold that is poised to captivate the scientific community. Goldene's sheer thinness, roughly 400 times thinner than the finest commercial gold leaf, endows it with remarkable optical and electronic characteristics. As light interacts with the sea of electrons on the surface of this gilded marvel, it can generate waves that concentrate and channel energy in intriguing ways. This phenomenon has already been harnessed in the development of gold-based photocatalysts, suggesting that Goldene could unlock new frontiers in fields like energy storage and conversion. The synthetic process behind Goldene is as ingenious as the material itself. Starting with a sandwich-like structure of silicon and titanium carbide, the researchers engineered a clever diffusion-based approach to swap the silicon for a single atomic layer of gold. The subsequent etching step, using a century-old Japanese technique, liberates the Goldene sheets, which can span up to 100 nanometers in width. While challenges remain, such as refining the separation of Goldene from the surrounding solution and growing larger flakes, the Linköping team's accomplishment has ignited a new wave of excitement in the scientific community. Renowned materials scientist Stefanie Reich of the Free University of Berlin hails the breakthrough, expressing her enthusiasm for the prospect of taking traditional metals and making them into really well-ordered 2D monolayers. As the search for novel 2D materials continues, Goldene's emergence stands as a testament to the boundless creativity of human ingenuity. This gilded marvel, with its captivating properties and promising applications, promises to usher in a new era of transformative discoveries. Source: www.nature.com. Alarming sinkage, China's major cities in rapid decline, sinking coastlines, a troubling trend in China's urban landscape. As the world grapples with the devastating impacts of climate change, a new scientific study has revealed a startling reality unfolding in China. Nearly half of the country's major cities are sinking, with some doing so at an alarming rate. The research, published in the prestigious journal Science, paints a sobering picture. According to the study, a staggering 16% of China's urban areas are sinking rapidly, at a rate exceeding 10 millimeters per year. An even larger swath, roughly 45% of the mapped regions, is experiencing a moderate rate of subsidence, greater than 3 millimeters annually. The affected cities span a diverse geographic landscape, from the coastal megalopolises to the inland hubs of the southwest. Even cities like Kunming, Nanning, and Guiyang, which are not as densely populated or industrialized as others, are grappling with this alarming issue. Subsidence is certainly not only a problem in China. Many other parts of the world share the same problem, says Ding Shaoli, a geodesist at the Hong Kong Polytechnic University. Indeed, the study notes that by 2040, almost one-fifth of the world's population is projected to be living on sinking land, with the Netherlands and Jakarta facing particularly severe challenges. The researchers attribute this alarming trend to a complex interplay of natural and human-driven factors, including groundwater depletion, the weight of buildings, and underground mining activities. Excessive groundwater extraction, in particular, has been identified as a key driver of land subsidence in cities across the globe. The implications of this phenomenon are nothing short of staggering. The study estimates that within a century, 1 in 10 residents of China's coastal cities could find themselves living below sea level, as a result of both land subsidence and the relentless rise of global sea levels. In fact, the researchers project that a quarter of China's coastal lands could slip below the waterline within just a few decades. This double whammy of sinking land and rising seas poses a grave threat to the hundreds of millions of people who call these vulnerable regions home. Flooding, infrastructure damage, and even entire communities being submerged are all very real possibilities. Yet, the study also offers a glimmer of hope. The experience of cities like Tokyo and Shanghai, which have successfully slowed their rates of subsidence through regulatory measures and environmental controls, demonstrates that with concerted action, this crisis can be mitigated. The key to addressing China's city subsidence could lie in the long-term, sustained control of groundwater extraction, the paper asserts. Dike systems and other adaptive measures could also play a crucial role in safeguarding these sinking coastlines. As the world watches with bated breath, the fate of China's urban centers hangs in the balance. The race is on to find solutions that can preserve the homes and livelihoods of millions, before the ground quite literally slips away beneath their feet. Source: www.nature.com